Thanks. So pi over, no, sorry.
Hey, that's mine.
I know. I'm stealing. I'm trying to get to mine. You should have kept going, Wayne. There we go. Hi. What I want to do today is give you a very different view of scale and a different view of science, I hope, which is a tall order for 15 minutes, but I'm going to try, and tell you how historians and anthropologists of science think both about science and scale and how science is actually done. The first book that I wrote, I co-wrote with a physicist, Herb Bernstein, and I'm happy to say on the cover, we were very happy to get this painting by Mark Tansey called Coastline Measure, and it gives you an idea of something I want to say. This was categorized as postmodern art now, and I want to talk a little bit about modern art in just a second, but what Tansey does is plays around with scale, and we use this as an image of what science, a kind of general image of what science looks like in this taking of measures. But as you know, if you notice that it appears to be on different, that people are working at different scales of things. And it suggests that maybe these things that we call science and ethics and art, we think of as happening in different places and maybe even at different scales. But if you notice how Tansey plays with scale here is that these are all, well, this laser pointer doesn't work very well either. It's a repeating fractal structure, and it's the same self-repeating scale down at each scale. And it's a nice image to suggest that maybe those things are happening all at the same time, that there's always science, ethics, and art all jammed together, and it happens at different scales, and I want to try to give you an image of what it might mean to think of science in this kind of way. So in order to start, let's not go from postmodern art, let's go to modern art. The biggest modern artist that there is, Pablo Picasso, who, if you were watching the news, you recently saw, sold the highest price painting in the world just in the last week. And Picasso really started modern art and the modern era that we live in, and this visual kind of imaginary where everything, all different stuff is kind of all jammed together. There's no one perspective, there's multiple perspectives. Stuff is coming in at weird angles. It doesn't really quite make sense, but it's huge. Nobody is huger than Picasso, right? But it wasn't always this way. In 1906, Picasso was painting very much in the non-modern classical style. This portrait of Gertrude Stein is it's flat, it's got a single perspective. Its main intent is to represent what the woman Gertrude Stein looked like, even if Pablo Picasso says it's a lie, it's the truth about Gertrude Stein, right? Just 1 year later, though, right, it all changes. The Demoiselles dAvignon, which hangs in the Museum of Modern Art in New York, where you can visit it, is the painting that is the breakthrough for Picasso. It's the first modern painting, and he cannot sell it for even $1 or a French franc, let alone $193 million, right? People hated it, they didn't understand it, but it was this new perspective, and it was a brand new perspective that only very few people would have understood, and those people would have been scientists, and one of those scientists would have been Albert Einstein, who in 1905 wrote one of 4 papers on the 4 major papers in this year of 1905 on the electrodynamics of moving bodies, which was a special relativity paper. Now, what do these two things have to do together? Well, they're both the introduction of modernism into 1 in art, and 2 in physics, but 3 into our world generally. The idea that perspectives can shift, that they multiply combine. This is not just the kind of made-up connection. This is actually a connection that the physicist and historian Arthur Miller makes in a beautiful book that's in the RPI library called Einstein and Picasso, Space-Time and the Beauty That Causes Havoc. And he shows how they both discard the empiricist view of what you see is what you get, and in favor of the realist intellectualist view, and thinking, not seeing, leads to truth, right? And they discard economical representation, and they try to get at this reality that lies hidden behind the appearances. And for Picasso and for Einstein, that was a 4-dimensional reality. And we know, because Miller shows, and he gets down at the very small scale of how Picasso and Einstein both work. that Picasso has in his studio these four-dimensional geometry books that he's learning in part from the French mathematician and physicist Henri Poincaré. See, he studied at very close level that kind of how can I project 4 dimensions onto 2? Right? And in the same way as Einstein, in the same year of 1907, is already starting to work towards his general relativity theory that won't be published until 1912, but he's starting to sketch it out. And in the same way that Picasso is sketching out, like, what would it mean to talk about, to present 4 dimensions on 2? Einstein is also trying to figure out how to think and how to present 4 dimensions in a 3-dimensional world of physics. All right? So, in many ways, and the argument of that book is a great one, but it's one that Einstein himself first made, that after a certain high level of technical skill is achieved, science and art tend to coalesce in aesthetics, plasticity, and form. The greatest scientists are always artists as well. And this kind of thing, that at the same time that you're doing science, at least at this very high level and a very theoretical level, there's an element of art to it. And you can't do science, you can't be a good scientist without also being a good or even a great artist. And this leads to the image of the good scientist as the daring and individual genius as well. Einstein is probably the icon of what it means to be a scientist. And he thought in terms of art. He also thought and admitted that there's no logical way to this kind of discovery, and there's only the way of intuition, helped by a feeling for the order lying behind the appearance. So he had that same kind of intuitive, a sense of there's something more to doing science than just the logic and the calculations and the math. Now, historians, again, are kind of impatient or at least quizzical about, well, intuition. I'm not sure Einstein would have used that word. Or feeling? I don't think so. But we can go back down the scales again to the manuscript. And what is the word that Einstein uses, at least in some of these occasions, which gets translated into English as intuition or feeling? It's Fingerspitzengefühl, right? Which is a great German word, which is a lot of fun to say. Say it now. Fingerspitzengefühl. Nice. Okay, sehr gut, yeah? Okay, what it meant was, it was from the German military, the generals were said to have Fingerspitzengefühl, fingertip feeling, if they could get a sense of the whole battlefield and everything that was going on all over the place, all at one time. And that's what Einstein said was necessary for doing science and doing science well. It gets translated into intuition sometimes, but it has a very different set of meanings in German. There's another expression for this. Anybody recognize this guy? Very nice, right? His Manhattan Project photo. Most people don't get the younger version of it, right? Feynman would say something similar. This is in an interview with the physicist Sam Schwaber in 1984. This is Feynman talking. The Feynman diagram is really, in a certain sense, the picture that comes from trying to clarify visualization, which is a half-assed, kind of vague, mixed with symbols. It's very difficult to explain because it is not clear. My atom, for example, when I think of an electron spin in an atom, I see an atom and I see a vector and a psi written somewhere, sort of, or mixed with it somehow, and an amplitude all mixed up with chi's. It's impossible to differentiate the symbols from the thing, but it is very visual. It's hard to believe it, but I see these things not as mathematical expressions, but a mixture of mathematical expression wrapped into and around in a vague way around the object. What I really am trying to do is to bring birth and clarity, which is really a half-assedly thought-out pictorial semi-vision thing. Okay? So art and science are hard to disentangle, and the scales get mashed up. Now, at another scale, let's go to the FBI. If you're a national security asset like Feynman and all the physicists were, the FBI at a very high level is monitoring everything that you do. And we know that when Feynman is doing these intense artistic scientific calculations in his head, he's also, as the appointee's wife, and the appointee is Feynman, was granted a divorce from him because of appointees constantly working calculus problems in his head as soon as awake, while driving car, sitting in living room, and so forth, and his one hobby was playing his African drums. His ex-wife reportedly testified that on several occasions when she unwittingly disturbed either his calculus or his drums, he flew into a violent rage, during which time he choked her, threw pieces of bric-a-brac about, and smashed the furniture. That's a different scale, right, than what's just outside his brain. And when you interrupt what's going inside his brain, you don't want to be around if you're a woman. Right? And that's a different talk. That's the gender politics talk. And neither Feynman nor Einstein nor Picasso come out very good on that talk. But let's go back to scale. So how historians think about scale, it's sedimented, it's compressed. We like these kind of geological metaphors or these geological images. So at this very meta level where it's hard to, you can't pin it down, it's just kind of everywhere, this Fingerspitzengefühl or the half-assedly thought out pictorial semi-vision thing is totally necessary for doing good science. Paradigms, theory, these big constructions about what makes sense, what explains something, down to the mesoscale where there are scientific organizations, communities, funding patterns, the changing technological instrumentation that allows you to ask different questions and see new things that you couldn't see before. The micro level of everyday practices that might be writing, that might be working with those instruments, and then down to the level of language and visualizations, what's happening there, what choice of word, whether you're going to use Fingerspitzengefühl or intuition, those kinds of very small, subtle differences might make a difference too, and then down to the level of data. We try to look at everything that's going on at these levels and see science, ethics, and art happening at all of them. So I'll give you an example, not from physics, moving from theoretical physics, but to the area that I study mostly now, which is in experimental genomics. This one chart captures a lot of what happened in the decade of the Human Genome Project, which, thanks a lot to government funding, drives the cost of sequencing down from $10,000 per million base pairs down to about $1 per million base pairs, and it's going to continue to go down. And at the same time, the amount of genetic sequence data that gets produced goes up exponentially, right? People thought all of this data will solve everything. We'll understand cancer, we'll understand asthma, we'll understand heart disease, we'll understand every condition that it's possible to understand. Didn't happen, right? Why doesn't it happen? Here's an article from January 2006 where it tries to describe what goes wrong with that vision of how data would solve everything for genome epidemiology. And I'll give you a sense of how this operates at all these different levels. At the pico level, at the data is going up, it's accumulating exponentially, but the situation is plagued with problems. This includes small, underpowered studies, often with flawed design. This is at the nano level of language, suboptimal conduct, biased analyses, selective reporting of positive results, lack of standardization among studies. Now we're getting up to the standardizing between platforms and between laboratories, poor reporting of results even from well-conducted studies, difficulties in assessing interactions, and so on and so on. The research evidence is fragmented. The interface between epidemiological and other biological evidence is poorly developed. So you can't make it all go together at these very much higher scales, and it's unclear how to keep track of the rapidly evolving evidence across fields that can be defined by disease, genes, or exposures, and how to rate the credibility of this evidence. Something else needs to happen. And it's not just for genomics. It has to do with cancer research. It has to do with fluorescent automated cell sorting, pharmacogenomic studies, drug development. They're all struggling with, we have all of this data, and it becomes difficult to know what's true. And in fact, all these different things possibly seem true, and then they sort of fall apart. It's a very modern problem, right? So, what are people trying to do? And indeed, there's an argument that can be made. John Unitas is a statistician in bioinformatics. He's a very talented man. Published this quite, in many ways, controversial article that indeed argues that most things that you read about in scientific journals are going to turn out to be false in the end, right? And in part because it's so easy to get good results that in the end don't really mean anything. But all right, so here's an example. In 2010, Science published this study of longevity in humans. They took 1,000 people who had lived over to the age of 100, just sequenced all of their genomes, analyzed the data to see, okay, what patterns can we see coming out? And they thought that they were able to predict, according to which of about 20 different genes that you had, or the mutations of those genes that you had, they thought that they could predict whether you would live to be over 100. And this was published in Science, in the advanced copy of Science, of genetic signatures of exceptional longevity in humans. What happened is that people began to look at that data, and now you're looking at all the variation in these genomes on the top red chart. There's a genomicist, Daniel MacArthur, who blogs at a place called Genomes Unzipped. He's like, I looked at this data, and look, those points are all over the place. And if you compare it, and you know, that's not the way that the data is supposed to look, but it got published in Science magazine. How did this happen? This is what a good genome-wide association study looks like. For coronary artery disease, Crohn's disease, rheumatoid arthritis, you get these very clean single peak signals, right? That one doesn't. That should not have ever been published, right? But it was, and although eventually, right, they figured out that what happened is in the changing from one generation of, and these are genome chips that have 660,000 markers in, so every experimental run you're generating 660,000 new data points, right? They found that in changing from one version of the chip to another, that lack of standardization screwed up their analysis. And once you accounted for the change at that level of the technology change, the whole study fell apart and ended up being retracted by Science. It never appeared in print. It only appeared on the advanced copy page. And this is a site that now monitors routinely the retractions that get taken in all branches of science. So again, what do we realize? This era of all this huge data poses new science ethical art challenges to people. And again, at these different levels, right? You can now collect human samples and sequence them to create huge amounts of genomic data, right? But biologists fail to design experiments properly. They do underpowered studies with insufficient sample size and then trumpet chance observations as biological effects. Here we're down, and the levels get kind of smashed together, but we're down at that very low scale of the level of data and the technologies that's used to generate it, right? But now we start to see the kind of social pressures that scientists will inevitably seek faster, more efficient ways to generate and analyze data because the field is so competitive. We're up now on the techno and the meso scale of your colleagues. Sloppy science comes from the pressure to generate surprising results and to publish them quickly, even though they are more likely to be driven by errors, right? And that pressure extends throughout the careers of scientists and is compounded by the preference of journals, including Nature and Science, to publish significant findings and of the media to report them. Now we're up at that kind of level of the culture where we love to see these exciting new findings about things like aging and so on, right? The cure, right, or the means to address this is to reform that practice of science ethics art to be more careful attention to methods, greater awareness of the potential pitfalls will help to cut down on the needless mistakes. It's an enormous uphill battle. There are people like Shirley Tillman, former president of Princeton University and a great mouse geneticist originally, Harold Varmus, former director of the National Institutes of Health and the National Cancer Institute, and others that are recognizing this, that want to rescue U.S. biomedical research from its systemic flaws, which have to do with these kinds of the competitive culture, this need for new findings, for producing exciting results that are going to advance your career rather than verifying prior discoveries. Right? So now we get this image of the good scientist not as the lone genius, but as the connected, careful, craftsperson who is focusing on every level of his or her experiments to see what can possibly go wrong here and how am I supposed to be thinking about this in ways that are difficult to imagine. And there's this kind of collective effort to make reproducibility a more vibrant and important part of the field. Now, why does this happen? All right, I'll give you one last. Once I got started on Einstein, I couldn't stop. There's a protein synthetic biologist named Hans-Georg Reinberger who turned historian and philosopher of the sciences. And he talks about experimental systems as this kind of labyrinth that you're constantly building around you even as you're trying to make your way through it. So if you imagine, and here's another dimensional problem where I have to do this in 2 dimensions, but you have to kind of imagine being down at that start line, right? And an experimental system can be compared to a labyrinth whose walls in the course of being erected in one and the same movement blind and guide the experimenter. In the step-by-step construction of a labyrinth, the existing walls limit and orient the direction of the walls to be added, right? The labyrinth that deserves the name is not planned and thus cannot be conquered by following a plan. We don't want a planned genomics. We don't want a planned science. But it's hard to know exactly how you're supposed to go and which turn you're supposed to make when you're inside the walls that you're also building at the same time. It forces us to move around by means and by virtue of checking out, of groping, of tâtonnement, which is another fine French word for that same kind of feeling of just like you're not knowing where you're going, but you're trying to move very carefully. Right? He who enters the labyrinth and does not forget to carry a thread along with him can always get back. And in retrospect, it appears, genius, Einstein, science. Right? But all along, you're just in these walls that you're trying to figure out the kind of way through. And you have to do it together. You can't do it by yourself. And again, since I got on that Einstein roll, Einstein knew that we're a pretty stupid species, right? But we're a little less stupid when we're careful and we do it all together and we think about all of these different scales at the same time. Thank you.